So this is just a quick overview of G proteins and how they're activated and inactivated within the cell. It is not in depth or anything, it is just a basic overview of the entire process. So basically G proteins are substances which have three subunits. You have an alpha subunit, as you can see here, a beta subunit, and a gamma subunit. The alpha subunit is basically able to attach itself to two types of substances. So you've got GDP, which is guanine diphosphate, or GTP, which is guanine triphosphate. So to decide on which one attaches to the alpha subunit, you have to look at whether it is inactive or active. Okay, so when the G protein is inactive, then GDP binds to the alpha subunit, or when the GDP binds to the alpha subunit, the entire thing is inactive, okay? And when GTP binds to the alpha subunit, then the whole thing is activated. The gamma subunit of the G protein is what attaches itself to the membrane receptor protein, and that is where G proteins are attached to the receptor proteins in the plasma membrane. So here's to give you an overview of the basic picture of where it is in the cell. Okay, so you've got the plasma membrane, and let's say that this is the intracellular fluid and that above it is the extracellular fluid. So you've got the receptor protein here with a ligand binding site. The ligand binding site is obviously specific to the ligand which would be able to bind to it. The phospholipid bilayer is just here, and it is a very simplified version of the phospholipid bilayer. In fact, you can't even see half of the things inside it, but oh well, it is just to give us an overview. And over here, inside the intracellular part of this entire schematic of sorts, you can find the G protein binding site on the receptor protein. So the gamma subunit binds to this receptor protein at this point, with the beta and the alpha just next to it, but the gamma is what is actually bound to it. And note here that GDP is bound to the alpha subunit. Why? Because the entire thing is inactive. So the G protein is inactive because the receptor protein is inactive because the ligand hasn't bound to it yet. Now let's look at the sequence of events to activate it. So this is the process of activation. So in the inactive form, you just have the ligand hovering over the receptor membrane, the membrane receptor protein. Okay, at this point it is inactive because it hasn't bound to it yet, and the GDP is still attached to the alpha subunit. So once the ligand actually attaches to the binding site on the receptor membrane protein, then the G protein detaches itself from the membrane protein because it undergoes conformational changes. The conformational changes also have another effect. They no longer make the alpha subunit amicable to binding with the GDP. Therefore, the GDP can no longer bind to the alpha subunit, and it detaches itself. In fact, it is replaced by GTP. Okay. At this point in the process, the G protein is not yet activated. It's still in the process of being activated. Okay. So because the GTP attaches to the alpha subunit, the alpha subunit goes through a teeny weeny bit more conformational change, which no longer enables itself to attach to the beta subunit of the G protein. Therefore, the alpha subunit with the GTP attached to it detaches itself from the G protein. From the rest of the G protein. So you've got the alpha with the GTP, and that is separate from the gamma and the beta subunits. At this point, it is active, and it can carry out various reactions throughout the body, throughout the cell, and it is a part of various signaling pathways and so on. Now to inactivate the G protein and get back to that stage up there on top. So the alpha subunit basically is able to phosphorylate the GTP which is attached to it. And GTP stands for guanine triphosphate. And when it is phosphorylated, you remove a phosphate group. Therefore, you end up with GDP attached to the alpha subunit instead of the GTP. And you have a phosphate group being removed. So at this point you have some sort of a weird reaction taking place like this. So you've still got the gamma and the beta, which are separate from the alpha, which now has a GDP attached to it, and a phosphate just being removed off it. Okay. At this point, the alpha subunit becomes, it regains the shape it once had, which can attach to the beta subunit. Therefore, it goes back and attaches itself to the beta subunit, and they can all go back and attach to the receptor protein on the membrane, or in the membrane, and the ligand can be removed, and the entire thing is inactive once again. So that is G proteins, a very simple overview of how they're activated and inactivated. There's a lot more to this, obviously, but this is just to keep it simple and just to give you a quick reminder of what happens.